So let's talk about inheritance. Here's the syllabus objectives. Right, another way that the syllabus requires us to be able to calculate the ratios is by, or express these ratios of the genotype and the phenotype ratios, is using a frequency histogram, which basically is a bar graph showing the frequency of particular characteristics seen in the sample. This is an example here of what we call a polygenic trait. As you know, human height is not binary. It's not just tall and short. We have a whole range, a whole spectrum of different heights. And that's because there's actually four autosomal genes responsible for determining height. And they can either be dominant or recessive. Now, the tallest people have dominant alleles for all of them, okay? The tallest people have 8 dominant alleles, and the shortest people have 0 dominant alleles. Okay, so the people in this side are going to be the tallest, and these people are going to be the shortest. And this is the ratio of the phenotypes expressed by the number of dominant alleles present, and this is the number of each of those. As you can see, it's kind of like a bell curve, and we can see that in a frequency histogram. So we've just transferred that information onto a histogram, and we can see that it's a bell curve. On the y-axis, we've got frequency of individuals with that phenotype. On the x-axis, we've got the number of alleles for the dominant trait present, so 8 down to 0. So just a few different types of inheritance. What we've talked about so far is complete dominance. So this is what this is this classic Mendelian inheritance, where we've got a single gene for a particular trait, and we have a dominant and we have a recessive allele. And so we see that the phenotype of the heterozygous state and of the dominant homozygous state are the same. So here I've got an example of PKU, which is a metabolic disorder, and it's a recessive, single gene recessive disorder, autosomal recessive. So it's only expressed when we have the homozygous recessive. So the phenotypes for the heterozygous, capital P, lowercase p, and the dominant homozygote, so capital P, capital B, the phenotypes are identical. No PKU. The phenotype is no PKU. There's one difference, though, of course, is that this one here is a carrier. It's a carrier of the lowercase defective PKU gene. This person won't experience PKU, but they are a carrier and they can pass on that recessive gene to their offspring. So that's complete dominance, and that's what we've been talking about so far. But there's also codominance. Now, I think of codominance as cow dominance because the classic example is for cattle, the color of the coat for cattle. So what is codominance, or the heterozygous genotype, displays the phenotype of both alleles of the single gene. So neither allele is dominant or recessive to the other. So the wild type is red. The wild type is the one that you see in nature, and the mutant is changed due to a mutation. So the wild type here is red coat, and the white coat is the mutant one. So in the homozygous red, only the red is expressed. In the homozygous white, only the white is expressed. In the heterozygous state, we have a roan color, where we see both the red and the white are expressed. So with codominance, we express that slightly differently. So we use the capital C for codominance, and the dominant alleles, we actually write a description of that. So R for red, W for white. So that's obviously different to how we do it for our complete dominance. So that's codominance. I think about cow dominance. And then we have intermediate dominance. So this is where we get a blend. Intermediate, incomplete, or partial, you might see. And this is the classic one we talk about with these flowers, where the red flower and the white flower in the heterozygous condition, they give you pink. So it's an intermediate phenotype, which is a blend of the other two. Now we have multiple alleles. So the ones we've looked at so far, there's only 2 alleles. But in some cases, there's actually more than 2. And a classic example of this is blood type. As you probably know, we've got A-type blood, we've got B-type blood, we've got AB blood, and we've got O blood. So let's have a look at how that works. It's controlled by more than 2 alleles. Any individual can only have 2 alleles, but there is multiple alleles. So there's 4 different phenotypes which are determined by 3 alleles. Okay, and it's an allele for a particular enzyme. And we express that with the letter I, capital letter I or lowercase I. So we have 2 dominant alleles, A and B. So both of these are capitalized because they're dominant. And we've got a lowercase I for the recessive allele. Now, so AB blood is an example of codominance because both A and B are expressed in the phenotype as AB blood. And both A and B are dominant over I. So let's have a look at how that looks. Okay, so we're actually going to do a cross between AB blood and A heterozygous. So what's that going to be? We're going to have a capital A, so I, capital A, but the other one is going to be the lowercase. So let's do this cross and we'll see what our genotypes and our phenotypes are. So in brackets, we've got the phenotype. So this person's going to have A-type blood. Equally, this here, okay, so this is the heterozygous condition. So we've got the dominant and we've got the recessive. And so the blood type is A as well. Now let's have a look at the cross on this side. We've got codominance here. So we've got AB blood. We have B-type blood. Okay, and so here's all of the different genotypes and phenotypes. The ABO blood type is a perfect example of multiple alleles. We've talked briefly about polygenic inheritance when we talked about height in humans. Another example is skin color. Phenotype varies along a continuum. It's not binary. And it happens when there's one characteristic that's controlled by 2 or more genes. And we looked at height controlled by 4 different genes. Grain color in wheat is another example of a polygenic type of inheritance. And then in this particular case, there's actually 3 genes that are involved in the color of wheat. And either they can all be either the recessive or the dominant allele, and the dominant allele is the one that gives the color red. So we have a pure white strain of wheat, which is all of the recessive alleles, and we've got pure red, which is all of the dominant alleles. So if we were to do a cross between these, we would come up with all of these different genotypes. And I've expressed the ratio of those genotypes in a histogram. And as you can see again, we've got that same bell curve. So what we've been looking at so far is inheritance of traits that are on the autosomal chromosomes. Now we're going to look at sex-linked inheritance. So that means that we're looking at genes that are on the X chromosome or the Y chromosome. And so we call them Y-linked genes for genes that are on the Y chromosome and X-linked genes for genes on the X chromosome. As you can see here, the X chromosome is much larger than the Y chromosome. So there's many more genes on the X chromosome, And the Y chromosome is really primarily related to sex determination. Remember, we talked about the SRY, sex determining region Y, and that's on the Y chromosome. Because males with an X and a Y chromosome, they've only got one X chromosome. And so because the males have only one X chromosome, whenever the allele is on the X chromosome, it's going to be expressed. Now, females have two X chromosomes, so only a recessive trait is going to be expressed in the homozygous. Males, whether it's dominant or recessive, it's going to be expressed because they've only got one. Females who are heterozygous may still be able to pass, or can still pass the recessive allele onto their offspring, and they're called carriers. So here's some examples of X-linked traits. So color blindness or color defective vision and hemophilia. Both of these conditions are far more common in males than in females. There's your syllabus objectives. And make sure you can, understand all of those. And here's the guidance. As always, the definitions are in the glossary. It says multiple allele inheritance. Refer to situations where there are more than two alleles considered incomplete and codominance. We've done those. Examples of patterns of inheritance could include hemophilia. We did that one. ABO blood for multiple. We did that. Grain, colour and wheat for polygenic. We did that. There was a lot there, wasn't there?